Hello aspirant. In this module, we are going to see the statistical concepts adopted in the bioequivalence studies. Bioequivalence studies are generally carried out for generic product development and for their regulatory approvals. Before going into the statistical concepts used in the bioequivalence studies, let us understand briefly about what is bioavailability and bioequivalence. Bioavailability is a measurement of the rate and extent to which a therapeutic active chemical is absorbed from a drug product into the systemic circulation and becomes available at the site of action. And what is bioequivalence? Bioequivalence is the comparison of bioavailability between two drug formulations. If two drugs are bioequivalent, there is no clinically significant difference in their bioavailability. Bioavailability studies are focused on determining the process and time by which a drug substance is released from the oral dosage form and moves to the site of action. On the other hand, bioequivalence studies are focused on the performance of the drug product and usually involve comparisons of two drug products, that is test and reference. Test product here is the generic product which is developed against the reference product which is already marketed in different countries. Bioequivalence is assessed by using two important pharmacokinetic parameters, that is the Cmax and the AUC. Cmax is defined by the maximum blood concentration observed in a plasma concentration versus time curve, and the area under the curve defined by the trapezoidal rule. Assessment of bioequivalence is based on the fundamental assumption that two products are equivalent when the rate and extent of absorption of test drug does not show a significant difference from the rate and extent of absorption of the reference drug when administered at the same molar dose under same experimental condition. Bioequivalence definition as per the Code of Federal Regulations by US FDA is that absence of a significant difference in the rate and extent to which the active ingredient or the active moiety in pharmaceutical equivalents or pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar condition in an appropriately designed study. Based on the Code of Federal Regulations definition, it is important to understand what is pharmaceutical alternatives and pharmaceutical equivalents. First, what is pharmaceutical alternatives? These pharmaceutical alternatives have the same active ingredient and they may differ in salt, ester, dosage form or strength. For example, you have a diclofenac potassium and diclofenac sodium. They are different salts, or these are available in different dosage forms like capsule or tablet, but at the same strength and same active ingredients. So these are all called pharmaceutical alternative. What is pharmaceutical equivalence? It is also having the same active ingredient and dosage form and strength. It may have the same USP standards like strength, quality, purity, and identity, and may differ in shape, color, excipients, release mechanism, packaging, labeling, and expiration date. The classical examples are generic drugs against the reference drug or reference listed drug. How will you conclude a bioequivalence study? There are different endpoints are used to conclude bioequivalence study, and these various endpoints are recommended by various regulatory agencies including US FDA, European Union, etc. First endpoint is the pharmacokinetic endpoint. So what is pharmacokinetic endpoint? It is the measurement of the drug in the biological fluids, that is nothing but plasma, serum, whole blood, urine, or fecal matters. So you will collect these biological fluids from the study participants who will be administered the study drug. Then as per the time points, you will collect the biological fluids and you measure the drug, and from that measurement of drug concentration in the biological fluids, you will assess the pharmacokinetic parameters. So this is called the pharmacokinetic endpoints, and these endpoints subject to statistical analysis which will conclude the bioequivalence. Then it comes to the pharmacodynamic endpoint. For example, the blood glucose level. You administer a drug and you are going to measure the blood glucose level, which is the action of the pharmacological action of the drug. So once you measure the blood glucose level, which will be the measurement will be subjected to the data analysis or statistical analysis, thereby you are concluding the bioequivalence. Then the clinical endpoint. What is clinical endpoint? It is the measurement of outcome of the drug. For example, you are applying the adapalene gel for treatment of acne vulgaris, or you are applying the diclofenac gel for the treatment of pain. So how much pain is reduced or how much acne vulgaris is reduced, which is the outcome of the drug, which is called the clinical endpoint. So it is typically a clinical trial. Then finally it comes to the in vitro endpoint. What are the in vitro endpoints? Some drugs which is not absorbed systemically, and those drugs will be acting in the local environment of the gastrointestinal tract. So those drugs are cholesterol or sucroferric, which will be bind to the bile acids or bind to the phosphates in the blood circulation or in the gastrointestinal tract. So these binding mechanisms will be exploited to understand the kinetic binding as well as the equilibrium binding. The constants will be derived and subject to the statistical analysis, and thereby you are concluding the bioequivalence. Finally, for the similarity of the formulation, you measure the physiochemical characteristics like pH, specific gravity, osmolality, surface tension, and viscosity, which will be compared between the test and reference product, based on which the equivalence will be derived. So these are all about the different endpoints used in the bioequivalence study. There are three types of bioequivalence, which is generally adopted by the regulatory agencies, and the most widely adopted bioequivalence is the average bioequivalence, which is the simplest form of bioequivalence approach, which is widely used by various regulatory agencies and widely adopted in the pharmaceutical industry for the generic product applications. So when it comes to the average bioequivalence, the average, the geometrical mean of the certain pharmacokinetic parameters will be considered for the bioequivalence purpose. Then it comes to the population bioequivalence and individual bioequivalence. These are the two bioequivalence approaches have been recently defined by the regulatory agencies, especially by the US FDA. So whether you take the average bioequivalence or population bioequivalence or individual bioequivalence, all types of bioequivalence will have a calculation of a criterion or a point estimate. So for example, in average bioequivalence, we have the test by reference ratio of the particular pharmacokinetic parameter, for example, Cmax. So you have test product Cmax and reference product Cmax. T by R ratio will be the point estimate. And then since it is done in a significant number of population, study population, for example, the bioequivalence study conducted in 10 subjects or 12 subjects or 24 subjects, you have multiple point estimates for each and every subjects. So you will calculate the interval for the criterion or the point estimate, and then you have a predetermined acceptability limit for the particular interval. So that is called in the average bioequivalence 80 to 125% or 80 to 120% in case of the non-log for transformed data. So you will have the point estimate, that is the criterion, then interval for the point estimate, then a predefined acceptability limit for the particular interval. So these are common for all the bioequivalence types: average bioequivalence, population bioequivalence, and individual bioequivalence. Now, what is average bioequivalence? Average bioequivalence involves the calculation of a 90% confidence interval for the ratio of the averages, that is geometrical means of the pharmacokinetic parameters, either Cmax or AUC, for the test and reference products. So this is calculation of 90% confidence interval for the ratio of the averages of the pharmacokinetic parameters. So for example, you have test and reference product Cmax values. So T by R will be calculated. Then for these ratios, the 90% confidence interval will be calculated from the mean how it is deviating. So that is what the calculation of average bioequivalence talks about. Then to establish the bioequivalence, the calculated confidence interval should fall within the bioequivalence limit of 80 to 125% for the ratio of the product averages. So this is how the average bioequivalence will be calculated. In this particular slide, we are talking about the BE assessment, whether it passes or fails, how it reflects. So here you can see how a passing BE will reflect the confidence interval distribution in the green color, and they are within the limit, and the variability is normal. Here when it fails, the variability is very high on the lower side, on the higher side. It is deviating from the geometric mean ratio on the minus side as well as the plus side. And you can see it is coming out of the limit on the lower side and coming out of the limit on the higher side. This is indicating the highly variable drug properties or highly variable nature of the drug product. So the bioequivalence decision is based on the confidence interval and the acceptance range. If the 90% confidence interval of the geometric mean for the Cmax and AUC within the limit means bioequivalence is proven. If it is not within the limit means bioequivalence is not proven or bioinequivalence is proven. This particular slide talks about why the bioequivalence limit is 80 to 125%. Why not 80 to 120%? It is sometimes 80 to 120% also when the data is not log transformed. But when it is log transformed, we used to keep 80 to 125%. Why? The regulatory bodies decided that differences in systemic drug exposure up to 20% are not clinically significant. So the appropriate range would be 80 to 120%, but that is not the range. This is because the pharmacokinetic parameters for exposure, AUC or Cmax, are log normally distributed, which means if we transform these parameters by taking the logarithm, we'll get a normal distribution. Thus, the symmetrical plus or minus 20% has to be in the log transformed phase so that the statistical test of bioequivalence will be valid. This particular slide talks more about why we keep 80 to 125% rather than 80 to 120% for a log transformed bioequivalence. So this table indicates the ln ratio for the 80% is around minus 0.223, and you have to keep the positive side also 0.223, but the 0.223 comes for the ratio of 125%. So when you keep balanced on the minus side also 0.223 and the positive side also 0.223, the ratio of the data on the minus side will be 80% and the positive side will be 125%. That's why when it comes to the ln ratio, we have to keep the 80 to 125%. 80% on the lower side will be minus 0.223 and 125% on the positive side will be the 0.223. So this is how we keep 80 to 125% for the confidence interval acceptance of the bioequivalence data. Now, can two generic products be interchanged? Although the assessment of average bioequivalence for generic approval has been in practice for years, it has some limitations. Many authors criticize that the assessment of average BE does not address the question of drug interchangeability. Though these questions are arises, still the average bioequivalence is the most widely accepted approach for the bioequivalence conclusion. Generic product can be used as a substitution of the reference product if their bioequivalence is demonstrated. Current regulations do not indicate that two generic products can be used interchangeably even if both of them are bioequivalent to the same reference product. Bioequivalence between generic product of a reference product is not required. Thus, one of the controversial issues is whether these approved generic drug products can be used safely and interchangeably. Theoretically, the potential difference between two generic products could be about twice as large as that is allowed between a generic and a reference product. The reason is that regulatory agencies worldwide require that reference drug to which the comparison is made should be the originator formulation. So basically, the acceptance of switching to generic product is generally not acceptable, and the bioequivalence is always proved with the originator formulation. Now, what is drug prescribability and drug switchability? Drug prescribability is defined as the physician's choice of prescribing a drug product for a patient who has not taken before the drug in any of its forms. The choice is between the brand name drug product and a number of generic drug products that have been shown to be bioequivalent to the brand name drug product. What is drug switchability? On the other hand, involves the switch from the drug product, either a brand name or a generic drug product, to an alternative formulation, again either a generic or a brand name product within the same subject, whose concentration of the drug product has been titrated to a steady, efficacious and safe level. So what here is drug prescribability and drug switchability defines that the pharmacist or the doctor have a choice. He has a brand name product which is the innovator product, and then he has a number of generic products which is bioequivalent to the branded product. This is called drug prescribability, where he has the options of prescribing the brand name product and generic products which is equivalent to the brand product, individually equivalent to the brand name product. Then drug switchability. Switchability is you are switching the brand, one brand to another brand, or one brand to a generic, or one generic to another generic. So this is called drug switchability to an alternative formulation for a subject who has been using this product for a certain period of time. This is called drug switchability. Now, what is the relation between the population and individual bioequivalence and drug switchability and prescribability? Average bioequivalence cannot imply either drug prescribability or drug switchability. Therefore, it is suggested that the assessment of bioequivalence should take into consideration of drug prescribability and drug switchability. To address drug interchangeability, it is recommended that population bioequivalence and individual bioequivalence be considered for testing both drug prescribability and switchability. The average bioequivalence method does not assess a subject by formulation interaction variance, that is, the variation in the average T and R difference among individuals. In contrast, population and individual B approaches include comparison of both averages and variances of the measure. The population B approach assesses total variability of the measure in the population. The individual bioequivalence approach assesses within subject variability for the test and reference products as well as the subject by formulation interaction. In case of population bioequivalence, to address the drug prescribability, FDA proposed the following one-sided criterion. So this is the formula used for the population bioequivalence criterion, where T and R are the mean of the test drug product and the reference drug product. Sigma 2TT and sigma 2TR are the total variance of the test product and the reference drug product. Here, multiple lots of the test and reference product will be used for the assessment of bioequivalence. Then the delta 2T0 is a constant that can be adjusted to control the probability of passing bioequivalence, and theta p is the bioequivalence limit for population bioequivalence. So basically, this is the formula is being used for the as a criterion for the population bioequivalence, and the population bioequivalence uses multiple lots of the test and reference product to understand the total variance of the test drug product as well as the reference drug product. This particular slide talks about the individual bioequivalence. So to address the drug switchability, the US FDA recommended the one-sided criterion for the individual bioequivalence. So the individual bioequivalence will be calculated by using this formula, where the micron T and micro are the mean of the test drug product and the reference drug product. Sigma 2WT and sigma 2W are the within subject variance of the test drug product and the reference drug product. So to understand the within subject variance of the test drug product and the reference drug product, it will be administered multiple times in the same subjects. And sigma 2D is the variance component due to subject by drug interaction. Sigma 2W0 is a constant that can be adjusted to control the probability of passing individual bioequivalence. Then finally the theta is the bioequivalence limit for the individual bioequivalence. So basically the individual bioequivalence talks about the drugs address that it is used for the drug switchability by the US FDA, and it uses the within subject variations by using the multiple administration of the test as well as reference product within the same subject. So friend, in this particular module we have seen the different statistical applications used for the bioequivalence approaches, different bioequivalence approaches like average bioequivalence, individual bioequivalence and population bioequivalence, and what is drug switchability and prescribability, and the relations between the different bioequivalence as well as the drug switchability and prescribability. So now moving forward will be see some other statistical application in the pharmaceutical development process. Thank you.